So, as already been introduced, I'm from the NIHR BioResource, and we're all about translational health research. So this is a quick summary of what I'll be talking about today, and I thought I'd better introduce the NIHR BioResource because a lot of you might not have heard of us. We are kind of the lesser-known sibling of the four big pillars of translational medicine in the UK. So you've been at this conference for a few days now, so you will not have failed to hear about UK Biobank. That was the first pillar, and all the amazing work that's come out of what UK Biobank have done with their 500,000 people. You'll have also heard a lot about Genomics England and the 100K project and the National Genome Research Library and all their work on rare disease and cancer. We are a similar organisation, so we're funded by basically the research arm of the NHS. So all of our work is about research that's going to be applicable to the NHS, but also other medical research all around the world. And so we have this national recallable cohort of over 350,000 people. And that's kind of what makes us different from the other people I've spoken about. The fourth one I didn't mention is our Future Health. They're up and coming. They're busy recruiting at the moment. So we have a recallable resource. When our participants sign up, we can contact them multiple times a year and say, there's this research study. Would you like to take part in it? So we can signpost people to clinical studies, or sometimes we can just use their samples and the data we've already collected to give to researchers. So we can recall people by phenotype, so their demographics. And we have quite wide demographics. We recruit all over the UK, so we've got all the ages, the genders. We can also give you information on the deprivation index of the people. So based on their postcode, where do they live, what might they have been exposed to. We've got clinical data if they're in one of our clinical themes. They do health and lifestyle questionnaires, so we've got self-declared answers on diabetes or family history or things like that. And then we layer onto that. This is what my team do. We do genotyping on most people as standard, but increasingly we're doing more and more whole genome sequencing, which is obviously why I've been invited today. So that's what we do. We bank samples and people and their data, and then researchers can come to us with their research questions, and if we can help them, then we will, by providing those samples or the data we've generated, or giving them access to the participants to generate things that we may not have. So that's what makes us different. So these are some of the themes we recruit to. We have quite a large healthy population, so if you're interested in controls, we have a big blood donor population that's really useful. We've got 50,000 people with inflammatory bowel disease. We've got about 40,000 people with a range of rare diseases. We've got a mental health bioresource, which I'll be talking about. This one was a bit different. This one recruits through social media rather than clinics, and so we have a saliva resource for this one. It's a lot harder to get blood, and that's going to be a bit of a theme of my talk, this difference between saliva and blood. Got non-alcoholic fatty liver disease, immune-mediated diseases. Of course, during COVID, we helped with that and have a COVID resource. And then the two of our newest themes are not really disease-focused. We've got our decipher theme, which is to do with children, because children are very different to adults. There have been some talks I've been to today showing that the methylation in children is different. You can't necessarily just downsize what you've learned from adults and use that for children. So we've been starting that resource. And another big theme at this conference is that you need diverse genomes for equitable healthcare. So we have our IBO, Improving Black Health Outcomes, where we're really focusing on recruiting more ethnic minorities into the bioresource and improving access to them. So my background was in rare diseases. That's kind of my passion. I know it is for a lot of this audience, and this slide's going to be really obvious to you. You know about this diagnostic odyssey and why we do this, why we need to help these patients. So our first project a number of years ago was short read sequencing, and as everyone's been finding, that was at that time solving 20 to 30% of our cases. Why are we not finding all of it? Big topic here. So we started a couple of projects, and the long read project is one of them. So But this long-read project I'm talking to you about, it goes beyond rare diseases. We are going to be sequencing, I'd say, up to 22,000 genomes. That's because, we've got the money for 22,000 flow cells, and a flow cell doesn't always equate to a genome. We have had to do some replicates as we've been learning, so I think we're going to get above 20,000 unique genomes. And we've got a rare disease subset, an eating disorder subset, and a more general population-based one. As Cora said in the introduction, people are crying out for these controls. So obviously, it's all on Oxford Nanopore. That's why I'm here today. We've done all the sequencing in 2 different labs in the UK, Cambridge and King's College London. This data's currently at the University of Cambridge Centre, but we are starting to work with Amazon Web Services. So I think when this comes out, as I've said it will, we want to make this available to the international community. Hopefully we'll be hosting that on Amazon and making it available in a nice way for you. So these 3 sub-projects we're working on, the biggest section up to 14,000 participants, are part of another research study that we started on genes and cognition. And we have an interest in this area because of cognitive decline and dementia. It's a big health topic. And it just so happened over the pandemic, when everyone was locked up, the researchers we were working with were ready to release this app to do cognitive profiling. So we emailed everybody in the BioResource and said, would you like to do this app? And over 30,000 people did do it. And it measures all sorts of interesting things, from reaction times and vocabulary and putting things in sequences. So there's 10 different varied tests that we've got these scores on. Great uptake over COVID when everyone had nothing better to do. And then a few years later, we've repeated that. So we've focused on the people who have 2 time points. So there's a couple of years between them for our sequencing effort. But I think this is also going to be a really great general population resource, because these people are from all over our bioresource themes. Then we've got an eating disorders cohort. This is part of our mental health cohort. So these are all saliva samples. And we're looking at the 2 extremes of eating disorders. So 2,000 with anorexia nervosa, so low BMIs, and 2,000 with binge eating with high BMIs. And I know the team there are really interested in repeats. So that's going to be an interesting topic. And then with rare diseases, we've got 4,000 undiagnosed cases that have had previous genetic screening. Sometimes they've got short read, sometimes it's whatever is the national standard for that rare disease. And within that, we've also got a set of 1,000 people with RNA-seq data, and we're generating mass spec proteomics data. So we're going to have a really comprehensive look for those 1,000 people. And of course, if we can, we'll be feeding back diagnostic results to their clinical care teams. So this has been a big undertaking, a massive learning experience for us. We started this back in 2023, and we had to learn how to scale this up, obviously with a lot of support from our Nanopore colleagues, and learning from the Singapore and the Emirati genome projects, those being the only other 2 at this scale at that time. I think what's different with what we're doing is that we are using real-world samples, we like to call them. We've been collecting samples for over 15 years in the BioResource. As I've said, we've got blood and saliva-derived DNAs. Some of them are nice and fresh. Some of them go back 15 years. They've been in and out of the freezers and used in all sorts of things. So it is a real range of sample quality that we are having to deal with on this. We're aiming for a 30x human genome coverage. I'll show you some data on that. We do the variant calling and alignment on the box. And we use a wash and reload protocol to try and get up to that 30x genome in one run. We're using Promethean 48s, and we run 24 flow cells per Promethean over 72 hours to get that 30x of data, which means with the 2 labs running 8 machines, we were making 384 30x genomes every week. So once we got going, we really plowed through. And then we do the post-alignment downstream off the boxes. So as I said, there was a lot to learn about this. The first issues were setting our goals, and I kept going to the bioinformatic team and going, look, guys, what N50 do you need us to reach in the lab? I have more of a lab background. What yields do we need to get? You need to tell me what we will do, and we'll see if we can make the samples do it and see if we can meet in the middle. And they kept going, well, no, Kathy, you just tell us what you want. And I'm going, but I need it to be driven by the downstream analysis. And we kept going around in circles until we eventually set this preferred 30x coverage. And ideally, if we can get it, we'd like all the N50s over 10, but that does depend on the samples. There was a lot of understanding the samples, the source, what extraction method they'd already been used with, and understanding when we should do fragmentation and how much. Because, of course, if you fragment too much, you ruin your N50s, and that's the power of this technology. But if you don't fragment, then your yields are low, and we're also finding yield is really important for calling the variants. One issue is ONT keep improving the technology, which is great. It just makes us really jealous because, we started a few years back, and we just can't recall everybody. The computing power to just use these new algorithms or change to the newest flow cells or the new things, it will introduce batch effects. So we've had to set our baseline and stick to it to at least get a data freeze across everybody once, and then maybe we'll rerun things in the future and improve. There was a lot of learning on which bits to do where do we use the power of the Promethean boxes, where do we use our own technology, how to make it run through and happen within the 72 hours, because that's really quite key for our throughput. And then because we were using two different sites, making sure those two sites were consistent, that we even tested different technicians and making sure that they weren't introducing biases. So there was quite a lot of technical replicates and benchmarking in there as well. And there was a lot of learning about how you set this up at scale. I now know an awful lot more about aircon and electricity than I thought I ever needed to. I must say, these machines, they pack out a lot of heat. If your aircon breaks and you've got 3, even 2 Prometheans in a contained room, it gets to 40 degrees in there, and your lab techs do not like working at 40 degrees, and to be honest, I don't think the Prometheans do either. So aircon is absolutely vital. We've also managed to blow circuits in the lab because of the amount we were drawing. We have issues in our building with the backup generator tests. You don't want the backup generator turning on in the middle of a sequencing run. That really upsets the bioinformaticians when they have to glue 2 different parts of the run together. We've even had some technicians who are a little bit more static than others, and them touching a flow cell whilst it's running is bad news. So grounding your lab techs is another learning experience there. But equally, when you want to keep running at the sort of rates we do, you need spare equipment. You don't want to be behind schedule because one little thing's broken, so having spare equipment. You make so much data with this as well. Having the data transfer, the networking to get this to your other compute nodes, the processing capacity. Now we've solved the lab issue and we're generating genomes so quickly, we've actually fallen behind with the rest of the pipeline at our data center. So keeping up with that's important. And then data storage. We had to make the decision not to keep the pod 5s. They're way too big. We are keeping it for a few hundred so that we can do some benchmarking in the future. But even just the rest of the data is eating up our storage area. As you're doing the processing, you make temporary files. We have to be really nimble about getting rid of those temporary files to make room for the next week's sequencing coming through. Automation's been really key to standardizing this, and particularly across the 2 labs. Mentioned about the sample quality already, and trying to make sure that with all this going on, we don't cross-contaminate samples, particularly with the wash and reload technique that we've had to adopt, and then making careful decisions about technology updates and installing patches. Security patches are really important, factoring them in around our really high throughput sequencing. So this is just a quick schedule of what the King's Lab was doing to get through 288 samples in a week. And the key thing we learned was for saliva to get the yields up, we had to do 2 washes and reloads. So the team always loads saliva on a Monday so they can do the wash and reload on Tuesday and Wednesday, and then they can do blood samples on a Thursday. They only need 1 wash and reload, and then they don't have to come in on a Saturday. So literally our schedule's built around a normal 5-day working week. And then we have the library preps on a Tech and Dream prep to feed all that loading of the Prometheans. So yes, we had five lab techs running this program, generating 288 samples a week on 6 Prometheans. So as I said, the processing, the downstream processing is way behind the lab work now. We're at over 20,000 flow cells run now, but our first data freeze is going to be about 5,700 participants. This is our current workflow, so we do the sequencing, the base calling, the alignment on the boxes, and then after that we're doing the epidemy pipeline we've selected those different modules for the different variant types, and we've just really recently added in the new Medaka tandem release. And then we do downstream analysis, we do lots of QC, and we've just been starting trying to make these genome level variant files that pull together these 5,000 odd samples in that first data freeze, so all those variants are all in one file. So as I said, we've got this difference in sample type across the cohort. So our eating disorders cohort is all saliva based. Our rare disease cohort is very blood-based, and there is actually 1 person at the moment with a saliva sample in there, but the scale doesn't really show that tiny bar. Genes and cognition, that's based about who in the bioresource did this app for us, and so a lot of them are in our blood donor cohort or our inflammatory bowel disease cohort, so they are blood cohorts, but quite a lot of them are in our mental health cohort, so we're getting some controls really for the saliva samples, and then we've got a small general population of, again, blood donor cohort in there. This is some of the initial benchmarking we did with the genome in a bottle sample, and one of the reasons we set our preferred depth of coverage at 30x or 90 gigs of data is because those SNPs weren't really starting to plateau until you got to 30x, and we saw this with our own sample data as well. So we want the 30x of the SNPs because we don't really want to be missing SNPs, although for some of these people we do actually have short readers as well. But other variant types did seem to plateau a little bit earlier than the 30x. Then this is some of the metrics we've got. So I've split it into blood and saliva. Underlying these, and this will explain some of the slightly weird shapes to the plots, there are multiple extraction methods in there. I didn't want to labour the extraction methods too much. We're stuck with them. They're what was collected, what was done at the time. We can't change that. I don't want to be giving people false impressions that one extraction method is better than another, because it's to do with the method, the lab that did it, the time they did it, and what's been happening to the samples afterwards. But you can see some samples in there are a little bit more degraded than others. And you can see there is a difference between saliva and blood DNAs, particularly in the N50s. We are getting a lot smaller N50s from the saliva set, and the quality is a little bit lower and the total depth of coverage is lower, but it's still good. This is another way of displaying that same data, and you really see that stark difference in the saliva N50s. So for our project, ideally, we would like all of those dots in that big top right quadrant where everything's above 10 KB N50s and 90 gigs of data. So we're a bit stuck with the saliva samples having the lower N50s, but we can get the data yields out of them. And the other thing to point out is N50 isn't the only measure of the fragment sizes you're getting, so you can look at the N10s and the N90s, and from this we can see that there are big fragments in there. So we know for our blood samples that there are some massive fragments. At least 10% of our fragments are more than 35 KB, and have been starting to look at some de novo assembly, and of course the bigger the fragments you've got and for phasing and things like that, the better. So there are big fragments in there. And even for our saliva set, where we know they're not as good quality, and maybe if we were collecting salivas knowing that they were going to be used for long read in the future, we would treat them differently and so on. I'm sure we can approve this data with, if you were set up for it. But even so, 90% of these saliva samples are above 1 kb fragments, and that's a lot better than short-read sequencing. And our first data freeze is using a lot of the samples that we were piloting. So things are improving as we've learned and we've gone into production phase. So in some ways, these are the worst of our samples. And you can see that about 85% of the blood samples hit that 30x genome coverage first time round, and more like 60% of the saliva ones. And then we are doing a top-up strategy for the ones that don't hit the 90 gigs. So this is where we go back, we use the same DNA if we've got enough left, and we barcode them, and depending on how much they were short of our preferred 90 gig, depends on whether they end up in a 2-plex, 4-plex, or 8-plex. So this is just a graph on a few hundred of the testing that the top-up strategy works, and you can see pretty much everybody that we tried. I think there was 1 or 2 outliers didn't quite make it above the 90 gigs there when we did the second time round in the merge. So although we're creating a data freeze, it will be improved over time as we do these top-ups. As I said, not everything will align to the human genome. The blood ones, it's pretty close to 99%, and I went to an interesting talk. Some of this might be, the stuff that isn't aligning could be EBV or other interesting stuff for people to data mine. But you can see the saliva samples, it's a lot more varied. Some of them are down to 70% human alignment, and this is the reason why. So when you work with real people and you send them a kit in the post and you give them instructions and you say, oh, please don't eat and drink before 30 minutes before you give a sample, this is what you get back. And these are the worst ones that all came in on a day. There were hundreds of these coming in at the time. And you can see people have been eating and drinking. We are not getting nice, pure human DNA out of this. So I think when you see some of those graphs I've shown and are about to show, you can do a lot with saliva DNA. And my point is, not everybody has access to blood samples. And if you want to do population scale stuff, saliva is a good way to go. You just have to be able to cope with this sort of stuff. So here's some of the stats. I was at Caroline Wright's talk a couple of days ago when she was telling us the stats for an average genome. Well, I'm hoping ours are looking really similar, but you can see there are some little differences between our blood and our saliva ones, but in general they're calling similar numbers of all the different variant types. So despite those differences, those shorter N50s and the lower yields, the saliva's performing pretty well in comparison, and I think we are hitting those standard genomes. We have been doing the distribution by ethnicity, so we've been using the 1,000 genomes reference. So in the first figure, the coloured dots are the 1,000 genomes reference, and the grey ones are our data freeze samples. And then in the second picture is us extrapolating on the different ethnicities onto our own samples, which is working quite well. We are still quite European biased, so it's about 92% in this data freeze. I was having problems visualizing it because the European contingent does blow the scale on any plotting method I tried, but there are other ethnicities in there, and actually our upcoming data freeze, the one after this, we've actually deliberately pushed the people with self-declared non-white ethnicity into the pipeline next. So we will be skewing the next data freeze towards our more ethnic participants. But even in the first ones, just randomly, this is showing that the bioresource is recruiting equitably across lots of genomes, all the different ethnicities that live in the UK. And that's what we want to continue. So it's there. And you can see in the saliva one, there's that massive female skew, and that's because eating disorders and even mental health disorders, people signing up for that project have a massive female skew. It's about 90% female. So that's just a feature of that cohort. Here are all those variant types by ancestry, and of course the African genomes show up with more variation, as you would expect. And then, of course, because we're interested in rare diseases, we wanted to prove this technology worked. So we did confirm in some of our previous cases that the SNPs and things were working. I'll just very quickly show you a nice compound het and a deletion. So we know for one of our rare disease groups, they have, in a lot of cases, one hit, and they know that won't explain the disease, so they're looking for compound hets. Obviously, in the past, they'd had to use trios, but with this technology, just the proband alone is good enough. So in this, you can see one of the alleles is derived from the father, one from the mother, and then in the proband, they've got both. So compound het is a big tick. We can detect that with this technology, and here's a nice, I think it's retinitis pigmentosa, big deletion that shows up really nicely in the data. Methylation works really nicely. This particular example's on the benchmarking set, but you can see in a typical gene that's, I think, to do with imprinting, you can see those different haplotypes coming out. So, we're really happy with the data quality that we're generating. I will just finish with one more slide about the differences between the blood and the saliva. So this is a new analysis to do with trying to deconvolute the signals within the samples. So one of my colleagues, he's taken some CPG sites from a publication, and we're assuming that blood cells are mostly going to be granular sites and that saliva cells should be epithelial cells. So we were expecting to see all the blood ones across the top like we see, but we were expecting the blue lines down the side vertically. But actually what we see is this diagonal line for the salivas, and we think that's because saliva is actually a mix of epithelial and white blood cells. So within that methylation signal, you're going to have to deconvolute for the different cell types if that's what you're interested in. But I think it's a really interesting sample set. So in summary, we are building this resource of genomes. We've shown that we can generate them at scale. We've confirmed that it shows up all the things you expect to see. It's going to be a really great tool for discovery, and we think this is going to be enhancing multiple health themes, and we're just really excited for when we can release this and let other people in and do discovery on it too. So thank you very much for listening. That's my team.